Hey everybody, welcome back to Biology Bytes. What if controlling HIV did not always mean taking medicine every day? That is a big question behind an early study led by HIV researcher Dr. Stephen Steeks at UCSF. Using engineered CRT cells developed by a nonprofit called Caring Cross. It's not a cure yet, and it's still early science, but the idea is pretty amazing. Instead of only giving drugs that block HIV, scientists are asking, what if you could train the immune system to fight back for a long time? But before we get into the futuristic science, let's talk about why HIV still matters. HIV is not just a biology topic, it's a medical story, a technology story, and honestly a human story. Modern treatments have changed HIV from a deadly disease into something many people can manage for decades. That's incredible. But many people still need ongoing treatment, regular access to healthcare, and lifelong monitoring. So a treatment that could last for years after one infusion would be a very big deal. To understand why, we first need to know what HIV actually is. HIV stands for Human Immunodeficiency Virus. That sounds like a lot, but here's a simple version. HIV is a virus that attacks the immune system, especially cells called CD4 T cells. CD4 T cells are like the immune system's group chat leaders. They help coordinate the response when something dangerous shows up. So HIV is especially sneaky because it doesn't just attack the body, it attacks the cells that help the body organize its defense. Now let's see how HIV pulls off this very rude biological heist. HIV has a step-by-step game plan. First it attaches to the CD4 T cell, then it gets inside. Once inside, it turns its genetic instructions into a form the human cell can understand. Then HIV inserts those instructions into the cell's DNA. After that, the cell can accidentally become a virus-making factory. So, basically, HIV breaks into the cell, slips its own code into the instruction manual, and from then on, the cell unknowingly helps build more copies of the virus. But one step in this process is especially weird, and it's one of the reasons why HIV is so interesting scientifically. Most of the time in biology, genetic information flows like this: DNA to RNA to protein. But HIV is different. HIV is a retrovirus, which means it starts with RNA and then copies that RNA into DNA. That backward step is done by an enzyme called reverse transcriptase. So, if normal biology is like reading a recipe from a cookbook, HIV brings a sticky note and then rewrites it into the cookbook itself. And once scientists understood HIV's steps, they could design medicines to block them. ART stands for antiretroviral therapy. These medicines do not usually work by magically deleting every hidden bit of HIV from the body. Instead, they block HIV from successfully making more copies of itself. Some drugs block HIV from entering cells. Some block reverse transcriptase. Some block HIV from inserting itself into the DNA. Others stop new virus particles from maturing properly. In other words, ART throws roadblocks all over HIV's game plan. And ART is really powerful, but there is one catch. ART can lower HIV to undetectable levels, which is a huge medical success. But HIV can also hide quietly in some long-lived cells. These hidden copies are called reservoirs. That means if treatment stops, HIV may come back because some infected cells were basically chilling in stealth mode. So, ART is kind of like the keeping security system on all day. It works really well, but if the hidden intruder is still somewhere in the house, scientists want to know if there's a way to deal with that too. That brings us to the immune system, and one of its most important players, the T cell. T cells are white blood cells that help protect your body. Some T cells help organize immune responses. Others help find and destroy infected cells. The problem is that HIV targets immune cells, especially CD4 T cells, which makes this battle extra complicated. It's like if the villain was going after the team captain first. Very dramatic and very unnecessary. Very HIV. So, instead of only trying to block the virus, scientists started asking a new question. What if we could take immune cells out of the body, upgrade them into a lab, and put them back inside? That's a basic idea behind some cell therapies. Scientists can collect a patient's T cells, give them new biological instructions, grow more of them, and then infuse them back into the body. Think of it like taking regular security guards, sending them to an elite virus detective training, and then sending them back on patrol. Now, the hope is that these engineered cells can help the immune system control HIV for much longer than a normal immune system could. But here's a really interesting part. Why would one infusion possibly take so long? Your immune system has something amazing called memory. That is why some immune responses can last a longer time. The body remembers what it has seen before it can respond faster the next time. With engineered T cells, scientists are hoping to create a kind of long-lasting immune patrol. The dream is not just block HIV today. The dream is teach the immune system to keep watching tomorrow, next month, and maybe even years from now. Still, the science is early, and researchers are still figuring out why some people respond better than others. Now, let's zoom out and connect this to the biology concepts hiding underneath the story. Even if you're not taking a biology class, this next part is really cool. In biology, we often learn that information flows from DNA to RNA to protein, but HIV flips part of that script. It starts with RNA and uses reverse transcriptase to make DNA. That makes HIV a great example of how biology has rules, but also exceptions that makes your brain go, wait, excuse me? HIV depends on enzymes like reverse transcriptase, integrase, and protease. Enzymes are proteins that help reactions happen by lowering the energy needed to activate the reaction. So, many HIV drugs work by blocking those enzymes. If the enzymes cannot do its job, HIV cannot complete its life cycle. It's like taking away the virus's copy machine, glue stick, and final assembly tools. Suddenly, the virus is way less productive. That sounds amazing, but we need to keep our science brain switched on. This is exciting, but it's not the time to say HIV has been cured. The study is still early. It involved a small number of people, and scientists need more data to know how safe it is, how long it lasts, and who it works best for. That's how science works. A promising result is not the finish line. It's sort of the next set of questions. And honestly, that's the part of science that makes it really fun. It really says the end. It usually says, okay, now explain this. So, what should we remember from all of this? Here's a big idea. ART is powerful because it blocks HIV from copying itself. The single infusion approach is different because it tries to train or reprogram the immune system to help control HIV over time. So, the story is not just about HIV. It's about how far biology has come. We are moving from medicines that block molecules to technologies that can redesign living cells. And that is your Biology Bite. Tiny cells, huge science, and one very big question for the future of medicine. Thanks a lot for joining us on today's video. We hope you had a lot of fun and learned something new. If you like this video, don't forget to like and subscribe for more Biology Bites. If you care about helping other kids learn, check out Pratham USA. They bring reading and learning to children across India and help build a better world. Visit prathamusa.org to see how you can help. See y'all next time. Bye!